Welcome to chapter 4. So today we get to talk about characteristics of cells and life. So all living things, which are single or multicellular, they're made of cells. And these cells, they're going to share some common characteristics. One is basic shape, and that would be spherical, cuboidal, or cylindrical, amongst others, right? There's an internal content, which would be cytoplasm, which I kind of call cellular glue and goo. It's kind of just glop, right? And this is surrounded by a membrane to keep it all intact. And then we also have DNA chromosomes, ribosomes, and metabolic capabilities. And there are two different types of basic cells. One is eukaryotic and the other one is prokaryotic. Eukaryotic cells, these would include animals, plants, fungi, and protists. These contain membrane-bound organelles, or kind of little mini organs, right? Organelles, I kind of in my brain immediately think of a mini organ. And each of these are compartmentalized in that cell glue, that cytoplasm, and they perform specific functions. They also contain double membrane-bound nuclei with DNA chromosomes. So that keeps the nucleus in a nuclei, right? So it's two little very organized things. Then we also have prokaryotic cells, and these are where we have bacteria and archaea. These are prokaryotic. They're kind of pre-cells, so they're not very evolved, right? And so they basically are just stuff sitting in a cell. So there's no nucleus or no other little organelles, membrane-bound baby little cells, okay? So looking at these different pictures, we can see why we need to study this stuff. So we have our genetics, right? And so this is for hereditary and DNA inheritance. We have color organization or cellular organization, sorry, metabolism, enzymes, reproductions, transport, etc. So a lot of fun things. So we keep talking about life, but what are the characteristics of life? Well, reproduction and hereditary are one of the big ones, right? So this is where the genome is composed of DNA that's packed in those chromosomes, and these can produce offspring either sexually or asexually. We also have growth and development, we have metabolism, and this is where we have chemical and physical life processes that make the body work. We have movement and or irritability, and this is where we're going to respond to either internal or external stimuli. And this contains self-propulsion of many organisms. So self-propulsion, to propel would mean to move. So self-propulsion would mean I can move myself because I'm saying self-move. We have cell support, protection, and storage mechanisms such as cell walls, vacuoles, granules, and inclusions. And we also have transports of nutrients and waste. So here is an overall picture of a bacterial cell. And you can see kind of the organisms. We have actin filaments, we have the chromosomes tightly packed, we have pili, which would be for motation, so movement, right? Inclusion bodies, flagellum, and that's like the main movement, etc. So this would be the overall structure of a bacterial cell. So when we talk about external structures, what do we mean? Well, these would be like appendages. And so my arms, my legs are appendages. And so for bacteria, there are two major groups of appendages. One would be ones that function in motility, okay? So this would be, for instance, flagella and axial filaments, like periplasmic flagella. And we also have attachments or channels, and these would be like your fimbriae or your pili. We also have glycocalyx, and this would be a surface coating. So those are your two external structures that we want you to focus on. So a flagella itself contains three parts. The first is the filament, and this is long, thin, helical structure that is composed of protein flagellin. And it can either be a hook, and it has a curved sheath, and it also has a basal body. And this is where there's a stack of rings that are firmly anchored in the cell wall. So here is a couple of pictures of different types of flagella. So you have your flagella filament in the middle, you have the hook on the outside, and you have different cell walls that contain your outer ring, your outer membrane, your basilar body, etc. And the flagella kind of whips like that. It rotates 360 degrees. So this functions in motility throughout, or of the cell throughout the environment. And so it can either run or tumble, and this causes it to move, and it can either be singular or multiple. We have 5 different types of flagellar arrangements, and these would be monotrichous, mono meaning one, so this would be a single flagellum at one end. We have lophotrichous, and this would be where we have small branches emerging from the same site. We have amphitrichous, which is where there's a flagella on both ends of the cells, and we have petrotrichous, and this is where there are flagella that are dispersed all over the surface of the cell. So here is one where we can see we just have a single flagella, right? This one has multiple flagella, flagella, and then this one has spirochetes multiple, right? So you can see that there are different arrangements. This one has a ton. So that's where we're going to look at and see what types of arrangements we have. We also have different flagellar responses, and this is where we can guide the bacteria in a direction in response to external stimulus, right? And so this would be a chemical stimuli, and this would stimulate chemotaxis, chemo meaning chemical, taxis meaning movement. And this would be both a positive and a negative. So it can move towards a chemical stimuli because it's a good thing, or it can move against it because it's a bad thing. So that would be the negative. You also have light stimuli, and a keyword for light would be photo, right? Photosynthesis, we're using light to generate energy. So phototaxis is photo, light, taxis, movement. So that's light movement or light stimuli. You also have signal sets in flagella that they move it into motion either clockwise or counterclockwise. And counterclockwise results in smoother linear directions, and this is called a run like we just saw. And then if you have clockwise, this would be called a tumble. So again, the tumble versus the run, and the different types of responses that we can have. This one would be, there's flagellar responses from no attractant and repellent, and it's just going to be tumbling, right, or running, depending on the different parts of it. And then you can also have a gradient. So we can see that we have lots of different types of movement. We also have periplasmic flagella, and this is where we have internal flagella that are enclosed within the space between the outer sheath and the cell wall. This is a peptoglycan, okay, is what it's called. And this produces cellular motility or movement by contracting and imparting a twisting or flexing motion, okay? So you can see the periplasmic here and how it can do a twisting motion. You have your outer sheath, etc. So let's take a moment and pause and see which of the following best describes the action of the prokaryotic flagella. So why don't you pause and come back? All right. So the prokaryotic flagellum, it is capable of doing all of the above. It can whip back and forth to move the cell, it can extend and contract to move the cell, and it can rotate to move the cell. And finally, it can also attach the environment and pull the cell. All right, great job.